Hello and welcome to today's webinar featuring Sébastien Jacquemont. I'm Greg Bousted from the Simons Foundation hosting today's session. We'll be getting started in just a moment, but first, know that we'll be fielding questions at the end of today's presentation. But you can ask them at any time during the session. To do so, just submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere, or if you have first obtained express written consent from the presenter to do so. Sébastien Jacquemont joins us today from Montreal, Canada, where he is Associate Professor of Pediatrics at the University of Montreal. Dr. Jacquemont is a medical geneticist and is also Associate Professor at the University of Pediatric Hospital at Saint-Justine. He holds a Canadian Research Chair, a Jean-Louis Lévesque Chair in Genetics of Neuropsychiatric Disorders, as well as a Swiss National Foundation Assistant Professorship. Dr. Jacquemont's work is focused on understanding the nature of cognitive, behavioral, and neural changes and underlying the association between genetic variants and psychiatric conditions. He studies copy number variants and in the chromosomal region of 16p112. His group is also active in clinical trials evaluating the efficacy of potential medications for Fragile X syndrome. We're grateful that Dr. Jacquemont has agreed to present some of his work today. Welcome, Professor Jacquemont.
Hello. Well, thank you for having me on this webinar. This is really exciting. It's very bizarre, but I'll try to do my best. I will be talking about genetics and neuropsychiatric disorders and especially trying to figure out how do the mutations lead to symptoms and then to diagnoses. So we will be talking a lot about copy number variants, not because they are better than other types of variants, it's just because they've been around for a little bit longer and we have more data, and our understanding of these variants is better than some of the other types of variants that we are starting to discover in autism. The other interesting thing with copy number variants is that they have, you can study gene dosage effects because some of them tend to be recurrent, so the same exact region can be deleted or duplicated. The other thing too is that a lot of these copy number variants do not seem to be associated with any compensation at the expression level, so duplication regions are overexpressed and the genes and deletions are underexpressed. So these copy number variants, we've been looking at them for several years now in the clinic, but through genotyping data that was generated for a GWAS analysis, you can also look at structural variation, and they are the variants that we've been interested in are rare, but cumulatively they're relatively frequent. And this is a study that was conducted by the group in Lausanne that we collaborated on, looking at copy number variants in the general population in the Estonian cohort in about 7,000 individuals. And what it shows is that these copy number variants that we see in the clinic are also present at a low frequency in the general population, and they are also associated with neurodevelopmental disorders. So this is a very simple outcome, which is here just the frequency of intellectual disabilities in these individuals of the general population. They were recruited in general practitioners' offices, and you can see that if you look at all variants above 250 kb, you already have almost 3% of the general population that's carrying one of these, with an odds ratio of about 3 for developing intellectual disabilities. So you go from 1.7, which is the frequency that you would expect, to 5.1. The other interesting thing that we'll be talking about too is that these variants, this effect is driven not exclusively but prominently by women. So this is the same data looking at a proxy measure of intelligence, which is educational attainment. And what you can see is that as the deletion increases in size, here you have an increase of lower education in the individuals who carry these deletions. And this signal is mostly driven by women in this cohort. So they seem to be the ones who are carrying these large rearrangements in the general population. And we're not suggesting that the men don't carry them, they're just maybe selected out of these types of cohorts. So this same approach had been conducted by the DECODE group, looking much more exclusively at copy number variants that were previously associated with autism or schizophrenia. So they really only took those CNVs that already had a significant replicated p-value with a substantial odds ratio. And what they showed too is that you lose about 1 standard deviation in your IQ, and then you also lose some skills in other cognitive tasks. And so these are all these blue measures here are the carriers in the general population who do not meet criteria for psychiatric diagnosis. And up here are the individuals who have a diagnosis of schizophrenia. So of course, this is an aggregate analysis, and everything's pooled together, but it gives you a general idea of what these variants are doing. So these are the different types of copy number variants that were observed, and I'm just going to jump to 10 here. What you can see is that for the most frequent copy number variants, and we'll be talking about 16p, you can start seeing a significant frequency. What you can also see is that the fecundity of these individuals with copy number variants is decreased, and it's more decreased in males than it is in females. And here you have this present for the 22q and the 16p, and you also have it at a nominal level for the 15q 1.2, which only has an odds ratio of 2. So this variant is very subtle. By the way, it's not associated with autism, but it is associated with schizophrenia. It has a very subtle effect on neurodevelopment, but it already has an impact on fecundity. And this is something that we see in the general population, but it's of course something that we see every day in the clinic. Whether you are a clinician in an autism clinic or in a more general neurodevelopmental clinic, you see just a lot more boys walking in the door than you see girls. And this is something that we've looked at also in aggregate in these neurodevelopmental cohorts, trying to figure out if there was any molecular signature of this gender bias. And what this study was showing is that as you select for deleterious mutations, so here you have all the copy number variants, and then here you're selecting for copy number variants that are deleterious, and then here even more deleterious based on their size, and then here you do the same thing looking at size as well as genes involved in the copy number variants. You see that the excess of genetic variants start to pick up in girls compared to boys, and this is compared to a very balanced number of copy number variants in boys and girls for the overall neutral ones. You find the exact same thing in the Simon simplex collection. At the time we were working on a much smaller segment of this, only 653 males and 100 females, and this has now been replicated in all the recent studies. And if you look at these variants and you look at how they're inherited from a mother or father, you can see that the inheritance is on average above 50%, so the 50-50 would be the random inheritance from either the mother or the father. And then as you increase the size and the deleteriousness of the variants, you start rising to about 60% of maternally inherited variants versus 40% of these variants being inherited by the father. And the Simon simplex in that reduced cohort, it was rising up to almost 70% of the variants being inherited by the fathers, and this has been replicated in more recent studies. So, looking at general effects of variants and general properties of copy number variants and single nucleotide variants is exciting, but we all know that there are, of course, general properties, but they also have a lot of specific effects. And if you start looking at specificity, then that's where, of course, the power starts being extremely problematic. So our group, as well as the assignments VIP, actually the assignments VIP started before us, focused on this variant, this region on the chromosome 16, which had at the time very interesting, was meeting different criteria which are really important for clinical research. First of all, it was relatively frequent, so it was frequent enough that we could make a cohort of this variant, and then also the mutation was recurrent. So the mutation that I'll be talking most about is the one that occurs between the breakpoint 4 and the breakpoint 5, and pretty much everyone who has this deletion or duplication has probably the same one. It's not exactly clear. There might be differences in the breakpoints, but we are not able to identify these differences unless you're conducting very complex molecular analyses. There are also other variants that we're interested in here, but they're not as frequent, and it's taking a little bit of time to gather a substantial amount of data on these. So you've already seen this slide. This region occurs either as a duplication or a deletion, and it allows you to study over or under expression of the genes that are comprised in this region.
Hey, Dr. Jacquemont, this is Greg Bausted from the Simons Foundation. We just have a quick question from
go ahead
here before we proceed any further, if that's okay. Sorry for the interruption.
So when you talk about one of these deletions, let's say, being inherited, do you actually mean Mendelian inherited? Let's say it was from the mother. She also had that deletion, or that it was a de novo mutation, but it happened to be on the mother's gene that was deleted?
No. So I'm actually talking about real inheritance. So not just So in the inheritance of the variants. So I didn't write it down here, but I think it's about half of the deletions occur de novo, or 1/3 of the deletions occur de novo, and or actually it's the other way around, maybe 2/3 occur de novo, and about 1/3 occurs as an inherited variant from one of the two parents. And then the duplication is more 1/3 occurs de novo, and 2/3 are inherited from a father or mother who carries the same duplication.
Okay, but when you say inherited, you mean the standard thing, and not...
Yeah, I mean the variant is, the mutation is present in one of the two parents.
Got it.
Thank you.
So the deletion and the duplication were identified in 2008, and so this is a seminal paper that got us all started on this research by Lori Weiss and the whole group. And we were at the time looking at something that was different, which was obesity in children who were referred for neurodevelopmental disorder. And we ran into a couple of these deletions in children who did have a developmental symptoms without autism and severe obesity. And several studies later, using several types of populations, general population cohorts, obesity cohorts, as well as a neurodevelopmental cohort, we characterize the contribution of this deletion and duplication in the alteration of BMI, and found that about a little bit under 1%, so 0.7% of individuals who have morbid obesity carry a deletion, And then if you start ascertaining kids or adults who have a neurodevelopmental symptom as well as severe obesity, the contribution of the deletion or the frequency of the deletion in this group is about 3%. And the relative risk for morbid obesity for the deletion is about 43-fold. So it's a pretty strong effect. And this association of neurodevelopmental symptoms and BMI control is not something that is specific to the deletion or to 16p, and in fact, we know rare variants that are associated with neurodevelopment and BMI issues. We know many of them, and in the general population, there's new studies, actually really exciting new methodology that allows to look for genetic correlation between human traits, and really confirms this at the general level, showing that there's probably a whole group of genes that simultaneously alter neurodevelopment, cognition and behavior, and alters core mechanisms involved in energy balance. So I think that this BMI or obesity could almost be a biomarker of a certain category of genes that fit this type of criteria. And then the last paper on genetic correlation by Bullock Sullivan, you can see the core. I mean, you can't see anything, of course, because this matrix is too small. But you see here BMI, and then the traits that are genetically correlated to BMI. And here you have a negative correlation with years of education. And this has been replicated recently in February by the Scottish group looking at IQ and BMI, and they also have a very strong, very remarkable correlation. So when we started looking at the 16p variant, we sort of first tackled the issue with a very traditional perspective of medical genetics, and that is what we used to do 15 years ago, or I don't know if I'm that old, but maybe 10 years ago. To understand the effect of a genetic variant, we would just gather patients who carried this variant, phenotype them, and then infer what the mutation is doing. So I think it's clear now with these new types of genetic variants that we are seeing now and detecting in our patients every day, and that have been detected also in the Simon simplex cohort. These variants have moderate effect size, and if you do this approach, you're not going to infer anything on what the variant is doing. So just by looking at a group of patients who carry the same variant with a moderate effect size, it doesn't tell you anything on what the variant is doing, because you're ascertaining probands with a very complex multidimensional problem, which is autism. Here I sort of symbolize this by 3 dimensions rising above a threshold. And the blue genetic variant here inherited from the father is contributing significantly to this problem, but it's only contributing maybe 30% or 40% to the overall problem. So if you want to study what this variant is doing, you have to extract the effect of the variant of interest from the rest of the genetic background, which is inherited from both parents. There could be some common variants here that I symbolized in green, I think, here because I'm colorblind, but I guess this is green. And then here maybe an additional rare variant coming from the mother here in red. And so we're not at the point where we can actually measure genetically these different contributions. So we have to do this indirectly by measuring the same dimensions in the proband and in the different members of the family. So without this design, and I think this is an issue now with some of the cohorts that were designed maybe 5 or 6 or 10 years ago, without this design of maybe less in the proband but the same thing in everyone, it's almost impossible to understand what the genetic variant is doing and how it's affecting quantitatively and specifically the 3 dimensions or one of the 3 dimensions. I think this is super important. And this relies on something that people knew a long time ago, of course, is that most of the cognitive traits that we're interested in and the cognitive dimensions have a very high heritability factor, and the heritability is the narrow sense heritability, that is, the variants that are contributing are really variants that are passed on from the parents. So here, for example, there is the example of IQ, which has an heritability of about 80%. You can infer the mutational burden present in the parent just by measuring the midparent IQ, and then you have a covariance of about 80%. So this is what we're doing in these families, and actually we weren't doing it at the very beginning, and we're definitely doing it now. And this is a very simple example for BMI, where you see the BMI in the family members, which is already higher than the general population, and then the BMI and the deletion carriers, which is shifted, and the BMI and the duplication carriers, which is shifted in the other direction. When you do that, what you see is that the effect of the del and the dupe is exactly balanced, and it's about 0.7 standard deviation. So it's a large effect size, but it's not large enough to rise above the rest of the genetic factors in here. And that's also probably why we're recruiting families with higher BMI, is that to get the child in the clinic, you have to have severe obesity, which is the effect of the deletion plus the effect of the inherited genetic background. Same thing for the head circumference. And I think the head circumference is this phenomenon with the head circumference is even generating more controversy in the field, because there's all this data on increased head circumference and increased brain volume and autism. But a lot of this increased brain volume is, of course, inherited by parents who do not meet criteria for autism. So you can't, parents are not the general population. They are individuals who already carry very specific variants that are contributing to the problem that you are ascertaining in the proband who's coming to the clinic. You see this for IQ. So these are sort of theoretical distributions of IQ that are separated by 25 points, general population here, and then a negative effect of a simple purely additive negative effect of minus 25 points. And this is the real data here of the deletion, showing that the effect of the deletion really is just on top of the rest of the genetic load, and there doesn't seem to be any particular interaction or epistatic effect. The group from David Ledbetter did this also, just looking at the Simon's VIP de novo cases with IQ, but also with the SRS, showing that the SRS can be, looks like it follows the same properties of a normally distributed trait in the general population, with genetic effects that are mostly working under an additive effect. So here you lose about 2 standard deviations if you add the deletion in the family. So this is the deletion and the duplication IQ summary from the latest paper by D'Angelo, which regroups all data from Europe and the VIP. And this is the intrafamilial controls. So you have a simple shift with a little bit of broadening of the distribution and the deletion, but in the duplication there is clearly other phenomenons on top of a simple additive effect. So it's unclear exactly if this is related to a bias, an ascertainment, or if there is a real genetic interaction going on. We've looked at the presence of additional genetic mutations in these carriers, and all deletion carriers carry an additional rare copy number variant that is known to be deleterious in about 3% of carriers. And if you look at duplication carriers, almost 10% of them carry an additional copy number variant that is known to be deleterious. And the difference between these two variants is significant. So, of course, there's not, I mean, we look a lot at IQ because most of the time in the cohorts, that's almost all we have. I think that tells you how, in a way, how phenotypically poor a lot of the cohorts are. I mean, IQ is a great measure, but it's just not going to get us to where we want to get. That is, trying to understand how the genetic variants contribute to a psychiatric diagnosis. So there are other measures. So phonology was well measured in the VIP and in Europe, and it also was well measured too in the Simon simplex. And what you can see is that the deletion and the duplication seem to have very specific effects on phonology. There's very specific alteration of phonology and language in the deletion, and the duplication is almost has preserved phonology. And I would even argue, even though I know a lot of my colleagues don't agree with me, but that the duplication, in a sense, people who have a duplication outperform in language individuals with similar IQ. So this is interesting because language is not really, well, used to be very important in autism, but is not necessarily an important criteria anymore. And here you have a variant that clearly is associated with autism and is also associated with poor language performances. And there's been a lot of literature to figure out whether specific language impairment and autism may have overlapping genetic factors, and I think that this is an argument in favor of this. On the other hand, if you look at the 1Q data, it seems that 1Q21.1, which also is very strongly associated with autism, doesn't seem to affect language at all. I mean, the effect size seems at least very small. So here you have two variants that both increase head circumference, both lead to autism, but one comes with a comorbidity of language impairment and the other one doesn't. So I think at the end of the day, this might start explaining why it's so difficult to figure out, or it might help us sort of decipher a lot of the heterogeneity and the subgroups that we're seeing in autism. You can do this, of course, with all sorts of other traits. The memory also seems to have a gene dosage effect and deletions, duplications with a clear outperformance of duplications who are very skilled in memory. Yeah. So I think that what these first studies are showing is that clearly these rare variants are very important and they're very useful for clinical research because you can really use them, I think, to start deconstructing the effect of the different genetic factors and their contribution to specific cognitive and behavioral dimensions, and how all this added together will lead to a multidimensional disorder, which is autism. But clearly these rare genetic variants are probably contributing in a minority of cases with psychiatric symptoms, and there's been now several papers on autism showing that the contribution to the overall variance of the rare CNVs and rare single nucleotide variants is actually even less than 10%. So that means that to really crack the whole phenomenon, we're going to have to start looking at rare variants in combination with the effect of common variants or maybe intermediate variants on specific cognitive and behavioral dimensions. I think this is a really fun proof of principle paper that came out of the Cardiff group. Actually, the Cardiff group also collaborated to the latest Doncheadle paper. And what they did is that they said, well, let's look at copy number variants, carriers of copy number variants who have schizophrenia, and they already carry a CNV that is highly associated with schizophrenia. So they already have a first very large hit that's putting them very close to diagnosis. What about the rest of the polygenic load in these individuals? And what they show is that this is the polygenic overall burden that can be measured today with new GCTA methods. Shows that schizophrenia versus control, you can account for about 10% of the variance of risk for schizophrenia. And if you start looking at schizophrenia versus those with the CNV carriers with schizophrenia versus control, they still also have a significant contribution of a common variance. And the same if you look at CNV carriers with schizophrenia versus the controls with the same CNVs, you see that there's a large difference in the polygenic risk score between these individuals who both carry the same CNVs, but one has schizophrenia and the other one does not meet criteria for the diagnosis. So here there's probably proof of principle that other variants that are very different from what we see in the rare variants, so other effects are present in our patients who carry rare disorders. And this is the same approach where if you start subtracting schizophrenia patients minus the ones who have schizophrenia and CNVs, you see absolutely no difference in the burden. So basically CNV carriers and non-CNV carriers, if you have a diagnosis of schizophrenia, you have to carry a certain amount of common variant burden. Oops, so here, okay, so, and this is data coming out from the Dosage paper showing that stratifying these 16P carriers between those who meet diagnosis for autism and those who do not meet diagnosis for autism. So this you've seen it before. It's the IQ of the deletion, the IQ of the duplication, and the controls. And then here it's stratified by the presence of autism. So if you look at deletion carriers without autism and deletion carriers with autism, there doesn't seem to be any difference. Actually, there's even a little increase of IQ that we're working on right now. But here, if you look at the duplication without autism and the duplication with autism, here there's a vast additional effect that is driving down IQ by almost 25 points. So really here, the duplication alone is very far from an autism diagnosis, and it requires a vast amount of additional factors to get it here. But interestingly, both of these variants have almost the exact same odds ratio, and their enrichment is similar in autism cohorts. But clearly, they are leading to very, very different types of autism. And I would even argue that the duplication may, in a sense, not really be an autism gene. I don't know exactly these individuals who have a diagnosis of autism in the context of an IQ of 30 or 40. It's a very particular type of ASD. So I think that this really shows you that maybe there is a very big difference between the autism risk between these two variants. So here what's also interesting is that we're actually quite interested in this little increase of IQ that you see in the deletion carriers who meet diagnosis for autism. And using the same strategy of genetic correlation, several papers now have shown that the polygenic risk score that was established in case control studies comparing autism versus control, if you look at what this polygenic risk score does in the general population, and if you look at covariance with IQ, you see that this polygenic risk score actually is associated with an increase of IQ in the general population. So it's quite interesting to see that all the rare variants that we know, not a single rare variant today that was discovered associated with autism, does not decrease IQ. So we have a very warped vision of a genetic risk for autism, which is rare variation associated with decrease in IQ. But if you look at common variation, it's actually the opposite. So you have probably genetic variants that are doing very, very different things and that are probably combined in the patients who have variants decreasing their IQ, and then on top of that, you may have some other variants that are increasing the IQ of these same individuals. And so I think this speaks towards this issue of pleiotropic, and I think there's been a lot of confusion in the field about what all these variants that we're working on are doing. And I think we thought initially that these variants were actually capable of having different effects in different individuals. And our working hypothesis today is that, in fact, these variants are doing have very specific effects on very specific dimensions, but combined together with other genetic background lead to different diagnoses. But this variant is not associated one individual with ID and then another individual has another effect driving ASD. No, it's always doing the same thing, but depending on the other additive effects, it's going to meet criteria for one of these disorders. So it's back to this issue of we really need to measure specific continuous traits on specific very valid dimensions in everyone in the family and really genotype everyone. It's going to take a little bit of time, and I think it almost calls for a new consortium of phenotyping rare variants in combination with the rest of the genetic background, but it's very essential, and I think it will be, in the end, very powerful approach. Also speaks for, interestingly, the specificity of these variants towards autism and schizophrenia risk. I think there's been a lot of discussion about this diametric effect on gene dosage. For example, the deletion of 16p is associated with autism, and the deletion and the duplication towards schizophrenia. And now that we have the autism data on the 16p duplication, I think this model is even more relevant because the duplication is really associated with a very specific type of autism, and I really think it's more of a schizophrenia gene rather than an autism gene. So this model is clearly still a very solid working hypothesis. So let's see. Running out of time. Of course, unfortunately, we would love to explore this, but there are some dimensions arising in autism, but in schizophrenia, we're still pretty far from some good dimensions. And today, nobody really measures the same dimensions in autism and schizophrenia, so it's hard to do really cross-disorder clinical research across these disorders today, but I think it's something that will come. Of course, you can look at neuroanatomy as a proxy because there's been a lot of neuroanatomical studies in both of these disorders. Simultaneously with the VIP, we have conducted MRI studies in 16p individuals, and this is a paper that was published last year showing that what you see in head circumference is something that holds up at the brain level. So the total intracranial volume, the total gray, and the total white matter volume are under a negative gene dosage effect. And now that we have more data, this is a very strong signal. If you look at the regional level, what you see as a very particular alteration covering the insula, the putamen, part of the caudate, the orbital frontal cortex, and the cingulate. So these regions that are under gene dosage show they're increasing in volume in the deletion, they're decreasing in volume in the duplication. This is corrected for total brain volume. These regions under gene dosage effect at the 16p region are not at all specific of 16p. This is something that has been seen over and over again in the autism as well as in the schizophrenia literature. And if you look at meta-analysis across autism and schizophrenia, and if you look at these top clusters that are picked, so these are the statistical maps that were picked up by gene dosage effects. If you look at these statistical maps and you look at their contribution in meta-analysis, you can see that autism and schizophrenia are converging a lot on these different areas. So putamen and insula are big alterations in these disorders. And this is even a more interesting study showing that if you do a regional analysis across psychiatric disorders, schizophrenia, bipolar, major depression, etc., substance abuse, you also see the insula, the cingulate, that are top hits across these disorders. So the 16p and maybe other regions, other genetic regions, might be so potent because they are already altering sort of a foundation, a neuroanatomical foundation for several different genetic disorders. So on top of this neuroanatomical alteration, you might just have to add a couple other alterations, and then you're shifting towards one diagnosis or another. So I think it'll be really interesting as we have more data on 1Q and other genetic variants that are also associated with autism and schizophrenia to see, can we start subtyping and clustering these very heterogeneous groups, such as autism and schizophrenia, into more neurobiologically relevant subgroups that converge on specific anatomical alterations. So that's going to be really exciting. But again, this calls for sort of a rare variant neuroimaging and phenotyping consortium. This is the same thing for 15Q in the Stephenson paper. So again, exactly the same regions, insula, cingulate. So there's several variants that are doing this. So of course the big problem is that, okay, so we spend a lot of money and time on 16p, but according to our good friends from Cold Spring Harbor and UCLA and Washington, there's probably 500 or maybe 1,000 genes involved in autism. So we're clearly not going to be able to do it the way we did it with 16p, and we have to be creative and figure out a way to collect data on all these variants and to run semi-aggregate studies to sort of flush out the signal that's common across groups of mutations, because we're not going to have the power to run individual cohorts across those 500 or 1,000 different genes. So I'm running out of time, but I'm just going to go really quick through trials. And it's a little bit pessimistic, but it's just the beginning, and just to show you the example of 20 years later when you're doing trials, the new problems that you're running into, and maybe what we might have to think about now to not replicate the errors that we did with Fragile X. So Fragile X, you all know, these are one of big intellectual disability gene that has a very strong association with autism, very well studied by many groups, showing that FMRP controls protein translation through a glutamate-mediated signal. And so all this preclinical data suggested that blocking this signal upstream of FMRP may help rescue some of the alterations found in the patients. And the trial that we did in Fragile X came after a very important substantial amount of preclinical data showing that multiple types of drugs can reverse phenotypes in mice, whether it's behavioral phenotypes or endophenotypes, structural phenotypes, synaptic alterations, you name it, everything. I mean, there was a lot of preclinical data. A lot of preclinical data looking also at different timings. So it worked in very young mice, but it also worked in adolescents and adult mice too. Maybe not as well, but there was a lot of data suggesting that it was even efficient in older individuals. So the idea was to maybe block it here, and we embarked on multiple trials starting in 2008, 2009, using drugs that are allosteric modulators that are blocking here this receptor and damping the signal that is upstream of FMRP. And the first proof of principle trials that were small but that came out in 2010 show that they were negative, but after a lot of post-hoc analysis, we actually found a really strong signal in those individuals who were fully methylated. So in a sense, super fragile X patients who had absolutely no residual protein levels. On the other hand, the ones who had partial methylation, so some residual protein, had a lot of different messy signal. And so here I'm just showing this because it tells you, once you've massaged statistically the data enough, you actually get really nice p-values. And this, of course, was published with a lot of caution that these were this is a pretty strong signal. And this is a replication study, so this is a massive study screening over 600 Fragile X patients. So these are very substantial efforts, and randomizing over 300 patients with Fragile X syndrome. And it took something like 24 sites over multiple countries and substantial funding by the pharma industry. And to make a long story very short, what we found is that all the preliminary data was incorrect, and basically there was no effect at all of the drug. The only effect that we were able to measure was that there was a little bit of secondary adverse events, more irritability and problems with behavior in higher doses of the drug. What you can see also is that we were able to measure substantial improvement in all of our patients. So we're really good clinicians, and the patients were really happy to come because they all improved, but the improvement was the same in the placebo and in the treatment. So the placebo run-in here, you're losing 15 points on your behavioral scale. So these are really massive effects. And this is an example of papers that came out. This is another trial, actually. This is the arbaclofen trial, but I think it speaks to the power of the placebo effects in all psychiatric trials. This is not specific to Fragile X. So psychiatry and non-psychiatry, somatic trials are associated with a lot of placebo effects. It's also possible that there's a very small subgroup of patients who actually do improve, but with the power we actually had the power to detect small subgroups of improvement, and we didn't see anything. What we see also is that there was absolutely no effect according to age. So we thought maybe that the younger we would go, maybe we'd start seeing a signal, but the signal isn't better in younger patients compared to older patients. So this is going to wrap it up. I think what's really important is that this molecular mechanism, which is controlled by FMRP, but is also controlled by other regions, ERK1 and TOR. This is a very complex mechanism that is controlled by many genes, and a lot of these genes are genes that we know very well. So CYFIP is in 15q, TSC1, tuberous sclerosis, and then 16p also. There's a recent paper that came out of Mark Baer's lab, and also the Roche group, showing that 16p also alters this exact same mechanism. So a lot of the new variants that we're interested in are going to alter this mechanism. So there's the same working hypothesis is going to be, we're going to have to test it again, and I think we're going to have to think a lot about what we do and how we do our future trials. I think there's a huge issue with windows of plasticity. How young do you have to run the trials before you can see improvement in cognition behavior? Behavior is also a general problem in psychiatry. almost no doctors who are in more modern fields work with behavior, but we're stuck with behavior. So I think the behavior measures that we have aren't great, but they have shown that they can improve in previous trials. So Risperdal shows behavioral improvement with the same ones we used. It's also been used in other types of trials. Cognition is clearly the way to go, but it's going to have to the trials are going to have to be longer and in younger patients to see cognition and cognitive improvement. So, yep, that's it. I'm just going to stop here. So I spoke on behalf of a lot of people. A lot of data I showed is a lot of consortiums. So I just want to thank everyone who works with us, really great collaborators. The students in the lab are really great too. And then for the Fragile X trials, of course, all the pharmaceutical support, and then all the collaborators who worked on the 24 sites for this huge, humongous trial. Thank you. Thank you very much for your attention.
Great. Thanks so much, Dr. Jacquemont. I think we still have enough time for a couple questions. We'll get to those in a minute. First, I'm going to check around here. Anyone here on site have any questions for Dr. Jacquemont? Okay, they're all saying it's crystal clear. So very good presentation. Okay, we do have a couple questions that were chatted in from the audience, so we'll get to some of those now. One comes from a postdoc at Case Western, and they want to know, these CNVs such as 16p11.2 are not on the X chromosome, so could it be that there are proteins with redundant function on the X chromosome that can reduce the phenotype in females?
I mean, the answer is yes, really. I think that males and females are different. It all boils down to the X chromosome. So the differences are there. I think it's just we thought initially that we knew those X-linked neurodevelopmental genes, but there are a lot of X-linked neurodevelopmental genes, but the ones that we know and the disorders that we know are just not. They don't contribute enough to explain what we're seeing. So it's going to have to be a lot of other probably X-linked contributions. Okay,
great. And let's see, another one here comes from a graduate student, a neuroscience graduate student at Johns Hopkins, and they want to know whether changes in head circumference, BMI, and 16p11.2 DELs and DUPEs are stronger at earlier ages, if that's something you've seen. And there's a follow-up question here. Which genes in this locus may be critical for these reciprocal effects? Any thoughts on that?
Yeah, so the head circumference increase and decrease in the duplication occurs in the first year and a half. So they're born pretty much with actually a slightly lower head circumference, and then it increases over the first year and a half. But then it stabilizes. It doesn't do the it's not the pattern that you see what's described overall in autism. So it doesn't normalize. It stays high. And the gene, so there is data using animal models, and namely it's using the zebrafish showing that KCTT13 with MAP kinase and MVP, the three genes together, probably are driving this brain volume phenomenon. I think that overall, what's really interesting is that the CNVs are almost little polygenic disorders in and of themselves. And so there hasn't been, in most of these recurrent CNVs, there hasn't been any rare variants that are associated with autism in one single gene. So it's possible that in 16p and then in a lot of the other recurrent variants, there's maybe 4 or 5 genes, and each of them have an effect size of maybe 0.2 or 0.3 standard deviation, and that's really hard to see on a case control study, at least with 2,000 patients. But you add 4 by 0.3 standard deviation, then you get a large effect. So I think that for a lot of these phenomena that we're seeing, it's already a little a little polygenic or oligogenic disorder in itself. And for the BMI, it's the same thing. I mean, we don't really have a clear BMI gene in there, and there might be several genes altering BMI in the same direction.
Okay, maybe let's see, maybe we'll try to get to one more of these. A listener wants to know, earlier you showed a slide that stratified the CMV effects by level of education. They want to know which countries were taken into consideration for this sample, and was wealth factor also considered?
Wealth?
Yeah, wealth.
Yeah. So it's this slide. So this is the work of Raymond and Catherine Manning. So education is a proxy of intelligence. So people use it when they don't have an IQ. So it's not as good because there's more environmental factors that are in there. So the heritability is not as strong also. And so this was done in Estonia. But the correlation between education and intelligence is sort of the same everywhere. It probably wasn't like this 200 years ago, where you could get an education if you were just, if you were just wealthy. But it's there now, and you could factor out wealth, but it's really complicated because intelligence drives education, which drives wealth. And it's very heritable. So your parents are going to be also intelligent. They're going to be rich. So it's a complex. You can decipher the effects out, but not with this cohort like this. So in this one, no, wealth is not pulled out. Phenomenon.
Okay, great. I think we're going to have to go ahead and end it there. If there are any questions that we didn't get to, feel free to post them during the replay. So there will be a complete replay of today's entire presentation available online by tomorrow at spectrumnews.org at the same site that the same page that you registered. And this is a monthly series, so check back for upcoming webinars. I think up next we have Niels Broos, all the way from Germany, presenting from Germany. I think that's an earlier start time, so check for the full schedule. That's on March 9th. And then thanks again, Dr. Jacquemont, and thank you all for tuning in.